describing a little bit about medical device design, which was called pufferfish. But in the corner of that pufferfish, we were also starting to build and design now automated microscopes. And have you connected it yet? Powered or not yet?
Not yet.
So we can all just move up here physically. And at that same time, if you want to bring that screen as close as possible and we can record. And the reason I want to show this.
Yeah, please feel free to stand up.
Yeah. Let's just come close. Was this the computer next to it or no?
Yes, the Ben said it works. Yeah.
Okay, so then try connecting that per se. And one of the things we're going to do, and the objective is missing. Yeah, so you can just connect that for a second and I'll drive it.
Oh.
So, what's important is at that time, I mean, of course, most of you all seen a Plaktoscope yet? Some of you have. And I think one thing we should do this next week, Kevin, is to get addresses for shipping Foldscopes to people that are online, but also just everybody in the class should have Foldscopes. So let's do that on Monday.
Okay.
And of course, manual microscopes are good, but there's a lot of times when you're trying to do something where automation is critical. You might want to say image a coral underground and underwater and you want to do that over 24 hours. And the focus is going to change because the weather is changing and the temperature is changing and something is suddenly moved. You want to be able to write code to interpret the data that you're collecting and feed it back. So suddenly actuators become important. Most microscopes nowadays are automated. And one of the things that we were starting to play with is, oh, wait a second, why doesn't there exist a modular platform to build any kind of microscope from scratch, from parts? And that sort of was the birth of SQUID. So if you open that PDF, for example, and in the meantime, I'm going to run, you running the SQUID code yet?
The main camera only.
No. We want to run it fully. No. Maybe what Ben meant was connecting that to just that. For this, we should be running the SQUID. Let's try SQUID tracking.
I think he said this one doesn't have tracking.
Yeah, that's okay. I'll just see as long as it has.
Did you say that is this the Raspberry Pi? Which other group has the Raspberry?
No, Plaktoscope has Raspberry Pi. And I'll mention this in 1 second. Let me just see which code do I want to run. Oh, it's not powered. Of course, it doesn't detect it.
There's the outlet.
So let's power things up a little bit. And then Hannah is on her way, and then she'll spend some time walking through and doing some demos for this. But on the history side, if you look at that PDF, you'll start seeing what we realize is that we started building these modules. And then these modules come together to build microscopes. The power? Yeah. See how far you need to go. Is there no other power here? Yeah, let's bring this one up. Funny, and it's supposedly a lab base. Oh yeah, unplug that, and then we will have a power cord. Okay, so how do you turn that into these modules turning into microscopes? The reason I'm spending some time on this is now all of these modules work. And for many projects, we can actually use these modules directly for automation, for control, for imaging, for cameras. There's a lot of time we spend. It's like, oh, where are all the cameras out there to give us some of the best? Okay, it's connected. Resolution for a given price point. And then we compared commercial, what are called scientific CMOSs, which are just glorified names for just regular CMOSs. But it's actually a security camera that beats all of it in field of view. So this is a D-hang camera, and I'm just going to show you the sensor for a second. What we ended up finding also is drivers are a massive pain for cameras and threads. So now we actually use this as the primary camera for all our imaging things in the lab. So I'll just show folks in here. This happens to be, and the reason they are so cost effective is because there is a mass production market for each one of these cameras. Because security has exploded in all kinds of ways, good or bad, whether you like it or not, has a consequence on the side. But they're also used in night security. So they have what is called the quantum efficiency, is how much photons can you convert into electrons. So as number of photons keep getting down and down, you need a high quantum efficiency camera. So, but now you can find lots of awesome cameras, and then you can spend your year trying to find drivers for it, because most of these companies don't release drivers. So it's also the fact that these had good drivers, people had written sets of drivers around. So that's one key component. If you're going to make a microscope or imaging systems, you start with this. Then the other set of a thread is a common microscope often is an objective. So you have an objective, of course. For Foldscope, we manufactured our own objective. But then on the other hand, there are commercial markets around objectives. And so this happens to be Boli. There are many different objective companies that primarily make objectives. We'll try to open this objective. And actually, let me ask you guys, can you guess how many optical components are in just this piece? Just only the objective. I'm not talking about this. Let's put 1, 5, 10, 100, 100, 10, 10, 5.
Well, not of all.
Okay.
Conform.
How many optical components exist in a traditional objective? If you were to really, and I'm about to open it up, when I say open it up, I'm going to open up the objective itself, which you should not do unless you know what you're doing. So I'm not going to open it completely, but so this is just a cover. This does nothing. Okay. This is just advertising. The actual objective is in there. And now you can see, I can see my set screws, and I could even start opening this up. If you did that, you will not have an objective anymore. Why is that? Why couldn't I just take the, so you can see sets of parts here, you can see sets of parts here, but stacked inside, if you were to cut a cross section, it depends on what you're trying to do with an objective. An objective could be trying to correct all the, so first of all, everybody remembers the laws of refraction. They're dependent on color, wavelength. So if I'm trying to make an objective that's going to be on a color camera, I need to have the same image form at the same point, but for all of these wavelengths. So color correction, and which is called a chromatic aberration, is actually super hard. So you cannot just use one lens. If you can use one lens, you'll make one clean image for one color. That's not what we do in microscopy. All of these colors coming, they would form blurred images for all the other colors. Similarly, your field of view, when you've written the fat lens equation and you solve the thin lens equation, we say these assumptions that only a portion of the objective is actually forming a flat image, because the rest of the focal plane is not in a plane. We have not as yet invented technologies that make these sensors curved.
Like
So
our
then,
eyeball.
like our eyeballs are curved. Does that make sense? So this is flat. It's not curved. Our eyeballs are curved. When you make lenses, you would then need, so then many of the objectives are actually correcting. It's called flat field, but you can only get flat field to a certain point. Then I can go on and on, you know, from fluorescence point of view. So that's the stack that adds up. So now if you just Google and search for, say, something, let's actually look what the answer is. It depends on the objective. So you're all right. There are objectives out there that have 100 optical lenses, and it's a single objective, and it's worth, if we had a chainsaw, which we will not do, you could actually cut this open as a slice, and what you would see is, so
When Anton van Leeuwenhoek was making his first microscopes, the problem wasn't getting a sufficient magnification. It was actually the aberrations that were affecting his ability to see the sample small.
and when we talk about Foldscope and optics in the Foldscope, we'll cover the differences between the two. But one of the threads is his insight was actually very clever. His insight was, this is called a compound lens, compound being many of them joined together. Leeuwenhoek made a simple lens because there was only one single lens at that time. But actually, compound lens microscopes were invented first. Simple lenses came later because of this aberration issue. Because with one single lens, it's very difficult to correct the aberration because mathematically you need more degrees of freedom. Every correction that we said required more surfaces to bend light. So now this is an example of what an objective would look like if we were to cut it. And that's why I said, never cut open your objective or disassemble it, because the magic and cost of making objectives. So this is the same object as this, by the way. So it's not something that, oh, it's very big. And now I can count, and somebody's sending me messages. Oh, is that you, Sonali? Sonali is not in the call, but Rupali is. Anyway, 1, 2, 3, and some of them you see stack back to back. You notice that this lens sits perfectly with this lens, and there is optical glue that binds many of them together. So these are many, many services. So let's count 1, 2, actually 10 might be the right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So this single objective, and you can tell that the way that it's aligned. So the problem in manufacturing lenses is not in making glass. We can make lots of glass. I can give you these 13, 14 lenses, but now align them with the same optical axis, all stacked together with really no errors in the lateral axis. That's what's hard. And that's really why objectives cost money. And that's why these things are built on templates. And still, the last time I visited a factory actually in India that makes objectives, you just sit there and people have a mastery. It's all still done by hand. When I visited Zeiss, there is a dark room in the basement. I mean, imagine these giant factories pumping out all of these things. You would say there would be all these robots. No, there are people in a dark room sitting who have honed the skill for 30, 40 years, because you align a little bit and you test it, and you align a little bit and you test it, and then you shim a little here, and they don't let anybody go in that room. And still, that's how we make objectives, because it's hard. Lots of people have tried many robotics, so you can get 90% there with robotics, and still every lens that most reputed lens makers make in microscopy is hand tested and is tuned. And so sometimes there are little, if this cover was removed, this was last put in, there would be these types of shims, and then they do interference patterns. So this is really why objectives cost so much money. So you can buy an objective that would be around $10,000, like one single objective, because of just the precision that was needed to get that level. So
How much? Oh,
sorry,
you go first.
man hour, it depends on how precise the objective is. And there is also become, because it's an art, people are like, oh, this is a Nikon objective or an Olympus objective. There has been, like, branding has happened. But I can tell you as a pipeline, the factory that was just making lots and lots of objectives, it's kind of like a Ford factory. You assemble this plant, then it assembles that plant, then it's so I think, I mean, in one objective, I would say it would probably take a day or two if there was just one person given all the optics to just assemble it and correct for it and test it and mount it. So, but this is important, but there is an industry out there for that, and there is a very large number of types of lenses. So this is another key component. But now, strangely, this and this also doesn't make a microscope. I have a camera, I have an objective, but it's not infinity corrected. What that means is the fact that the image that's coming in is not something that you can capture on a flat surface. Infinity corrected is how I'm seeing the world. All light, I'm assuming, is coming flat to my eye, and then I have optics in that that can map to the sensor. So one of the things that we often do is that there is a telescope. Also, it's possible, what I mean by a telescope is just something that takes an image that's coming in with a certain size, but it will form a tiny image on this sensor, and I'm not using all my sensor. That would be wasted. So does that make sense? That an image that might be coming out of this objective on the other back focal side is not the same size as the sensor. So I need to either stretch it or expand it. And so this is what we often use to do something like that. And this, the general optical term in this is a telescope, but a telescope can have 1-to-1 magnification. It takes parallel light coming in, or sometimes non-parallel light coming in, and makes it parallel so that it can be on a sensor, but it optimizes. So this is where, and then again, there is also an iris in here. So the set of an iris will allow us to control the amount of light. There is also a degree of freedom that you can choose that'll allow you to change the distances associated. And then I'll let you see the iris for a second. So I'm opening and closing that iris. And why would you want to have an iris in a microscope? Do you see that?
Yeah.
It closes. So you all can watch it right there and there. It's a mechanical object that's moving in and out. And then intuitively, where would you want to put the iris? We don't see often, you want to put the iris in a kind of a back focal plane, another point where there is convergence. Because then, one of the things you're trying to do is you're not trying to cut off an image. With the iris, you're controlling kind of intensities of lights and kind of how much light can enter in your optic. So there are very strategic places you put irises. Iris is super hard to make, but they're manufactured and they have all these flexure mechanisms of plates coming together. There was a project in some time ago of just, could we make a fluidic iris? And our eye is a kind of you can think about how your iris and your eye works. Yes.
Let's guess. One of the questions before was, how much did you account for the optical glue when you put the two?
Absolutely, super important. So this object has a ton of optical glue. And the way you use the optical glue, we have some optical glue, by the way, as you start playing together, which has a known refractive index. And there is a key trick that you use. It's UV curable. Because you have that moment you've assembled your optical system, and even if you sneeze on it, it's going to go back out of it. So you freeze it by using a UV curable glue. And so Norland is this big company. We have that in our machine shop. If any of you is doing very precision assembly, you kind of just align it and flash of light, and in UV it cures. So it kind of freezes in place. So that's kind of the purpose of optical glue. Now, many of these things have glues that are also in between on the corners. That's why you cannot disassemble some optical components because they get destroyed. Okay, so we found these sets of things and we found cameras, and then we started realizing that, okay, now this becomes the first core of a microscope, but it's still not a microscope because I would just be moving it around. I don't have any control. Then we started building these modules. And again, the whole purpose of SQUID as a paper, if you all spend time reading that paper, you'll realize that there are many hidden modules. So one quick module I want to show you is then we got started on designing and driving linear actuators. This is one we ended up choosing. And again, this is all open. It's all described. You can see it has both the motor, but then that's the linear actuator. You can see I'm spinning and something is moving, right? Right there. So now, finally, if I have 3 of these, I can do X, Y, and Z. That's exactly what's here. We just machine this little plate, and there's little bearings in here. You can spend some time, look at this. So another motor, the motor is just right there. You can actually see it's one of these NEMA motors. You can even see the plates. You see these plates right there?
Mhm.
You can disassemble and remove this motor and put another motor, but now you have to convert this motor into a linear bearing. So this is a linear bearing that allows you to take the shaft of this rotary motion into what you care about most, which is linear motion.
Mhm.
Normal motor, when you turn, doesn't actually have the shaft move forward, right? I mean, motors just spin, they
Yeah,
don't.
yeah, yeah.
So you can feel it that there is actually a helical track inside. That helical track converts rotary motion to linear motion. And then you ask yourself, oh, now I have this. How do I couple it to a stage? So one of the things is you'll notice that these stages are spring loaded. Because otherwise, if you lose contact, then you're not controlling how much you move. And there is a range to it. There's all kinds of threads. There are tricks around how to make these types of contact. So now we have, and you can see that there is another stage, but then can you identify there's something very special mounted on this? We just glued a piece. There is that golden piece that's only added on the Z stage. What could that be? Like it's almost as if a brick of
It
gold.
looks like it's like multiple
Yeah,
bricks of
can you
gold.
show it to the camera? So everything is good. This motor is very similar to this motor, which is on these two parts. But for Z, we added a special component here, which is this tiny gold block. It's not gold, so yeah. So for what do you need in Z axis that's different in a microscope that you don't need in X and Y?
Don't you also need to like connect back to the actual optical component?
Yeah, the optical component is mounted here. So you can see that in this sort of a context, this particular design, the stage is moving and the optics is frozen. Sometimes we do stage separate and we mount this on the optics. So there's different sets of design. Usually I like and prefer that we mount, we decouple the two where this is not on top of this. Actually, the design, this is Enrico just put something together. The original design, this mounts on this, and this is on a separate post. But he's just come to the lab, so he's assembling these for the first time. But this is also cool. Like he figured out a different combo of just putting it together. In one afternoon, when I had told him to assemble it, he actually assembled it differently, which is nice. Like you can start seeing the modular part. So that's a piezo. Because Z-axis, it's very sensitive to focus, and you need to move nanometer steps. And the piezos allow that super cheap component. If you need nanometer scale precision, tens of meters, that can be done there. There are drivers that are associated with piezo because they're high voltage drivers, but there are commercially available drivers. Yes.
Sorry for this. So the little linear motor driven actuator there.
We'll do the
plus-
macro
I
plus.
see.
So
Okay.
if I want to move 20 microns and 100 nanometers, you get close to the 20 micron with the macro motor, and then you add the 100 nanometer on the piezo, which is a voltage on a ceramic block.
Gotcha.
Yeah.
Piezo, you said piezos are cheap? I thought piezos were expensive.
Piezos are super cheap. It's the controller for the piezo
Okay.
that's expensive.
Really?
And it's just this strange thing. I can buy for 10 cents a piezo material.
Okay.
But it's the electronics, and it's high voltage electronics. And actually, this has been another hidden secret that it's not so hard. So there's lots of open source drivers. It's just high voltage. So companies have sort of made this. And
I see.
companies like the hats that you're hearing that
It's
will
a
not
free
be
hat.
named.
I'm not like advertising.
No, we use a ton of Thor Lab parts. We love Thor Labs. Okay, I have to run, so I'm just going to say the last set of a component. These posts, you can see they're Thor Labs. You can machine them, breadboards and sets of components. You can see those. So now you're starting to say, oh, I have a microscope, right? But the core vision of SQUID, now it's expanded to 30, 37 different microscopes. So if you go, now we have commercialized this platform. So if you go to cephla.com, you find all kinds. I mean, we make confocals, we make optical pool screening, expansion any high-end microscopy you can imagine. But at the core of it, it's always this. And it's open. Then the hardest part of all of this was the sets of drivers and a completely open source software stack, which Hannah is gonna do a quick demo for. I have to run to start the group meeting. No, just make any sample, just spit on a slide. There's, I can give you a slide, yeah. But one of the threads around this is it's a Python stack, and I'll talk about that in the class separately, because some of you can realize that, oh, I'm getting for free 5 different motor control, all the light control, trigger control, time lapse. Like, how do I fork the SQUID code and build it into? That's the point. The purpose I'm spending some time on these projects is we can fork them and build new things with it. So you don't have to then learn, and you should learn, but sometimes in the undisciplined learning approach is you want to get something working so you're hooked onto it. So also there is a Qt Py stack for the visualizations. So the entire engine, there is a visualization engine written on it. It has a UI. So using these sets of projects, and depending on people as they're choosing their projects, if automation becomes a key part of it, that becomes a valuable source of thinking about, oh, I have this entire tech stack accessible. All of the code and the project is on GitHub, so you can find the sets of files. And then later on, when we do a little bit of circuit design, I'll talk about this. So Arduino, you can see, is a classic microcontroller for all kinds of projects. It's really worth it for you to spend the time and learn. This has totally changed the game. There's also, you can read about the history of this project. There's a lot of messiness. The founders that they started it, they split. So there was a lot of debates going on and thinking about how to have a good open source project started. But then it is on a driver stack, and the drivers primarily have what are called edge bridges or sets of motor drivers. So there are 3 drivers in there for these particular motors, and there are specific chips that exist. So if you do a Google search for the data sheet for this motor, you will find a corresponding chip that you can buy that already has the driver stack written on it. So then it becomes much, much easier. So often enough, and then this board is basically a split out board. It has more number of parts, so we can actually operate 4 actuators with it. We can put inputs in, we can put light. So when you design these driver stack, we design them for many things. And actually the first version of this driver stack we designed was for a ventilator, ICU ventilator. And then we're like, oh, we don't need to do 2 things. Let's merge the driver stacks. And actually still in the lab, many other instruments that we make utilize this driver stack. Okay, on that note, I have to run. There is a camera cord. You could just swap this with this itself. There is a cord right in there. And I think when I had Enrico tested this, and this is up and running basically. And play with it. If you can't get it up and running, I'll kind of do that on Monday. But this was important to kind of get you the sense. What I want you to remember is everything that you see is modules or Legos all the way down. And so you should not just be thinking about a final object because, yeah, you know, I'm not working on a microscopy project. Why do I need this? But what you're working on is sets of things that are modules and are present that can then be incorporated in many other projects. Okay, I'm going to run for my group meeting. You're in good hands. Play around, ask lots of questions, post on Discord. If other people can't answer, I'll try to get to them. And then Ethan is going to come and do a quick demo also on the Plaktoscope. But instead of the demo, ask him to walk through all the sets of parts and a little bit around kind of these sets of conversations. Should just be much more driven by questions and ideas and conversations. Because these objects exist, and for certain sets of projects, we can carve some of them out dedicated to, because we make many, many, many of them. In terms of cost in parts, that's important to discuss. Something like this costs us roughly between 3 or 4K for us to make. We are now starting to shrink that cost further, but it's important that we also keep the modularity. We keep these modularity primarily because then it's easy to reconfigure. When you turn something into a product, you can further reduce the cost, but it loses some amount of reconfigurability. So when we productize them, we make them very differently. This is really just plug and play that you should feel that, oh, I can hack this microscope.
Sorry,
Yeah.
I know you gotta go, but
Yeah.
what is just the bulk of that 3 to 4K? Like,
Hello.
is it just all of
It's
it adds up?
scrambled together
Yes.
stages. SQUID has the
It has a
BOM.
Gotcha.
And that BOM has expanded, so when I'm saying 3-4K, that has fluorescence included in it, it's like a filters are super expensive.
Nice.
If somebody can just figure out to make cheap filters, like high quality chroma filters, you would give chroma a run for their money, quite literally. The laptop always ends up being a big chunk
Oh,
of
yeah.
like 1K or so, because we do some ML models on them. We like gaming laptops, you don't want to be stuck in. But just take a look closely. I think the whole point of this is a prelude. So take every little detail that I mentioned and things, and 1,000 times more is in that PDF. Okay, I gotta run my GM. All right, I'll
Thank you.
catch you guys.
Yeah.
Thanks, Mana.
Yeah.
Alright.
See.
And then, you can get it up and that's great. Don't worry about it. Just talk
Yeah,
a
for
little bit
sure.
about the adaptation that you are doing.
Yeah, for
Just so people get a sense of what happening these systems. And I'll see you.
yeah, it's happening now. Oh, were you gonna say that?
No, it just like he put a lot of information out there. If you have any questions about like what a driver is, or like you want to go more into the optics, like we can break it down a little more thoroughly. Yeah.
Yeah, this can also just be a general Q&A. So I don't know where you guys, this is like, y'all have projects, right?
Yeah,
Yeah,
nominally.
no, okay.
On paper.
So I was gonna say, so we can do whatever would be most useful for you guys during this time. So I'm happy to do a demo, and then if it would be helpful for you to have project work time, or if you would rather keep messing around with this, like whatever
Demo sounds great.
y'all are more interested in. Okay, so I do not have a sample. I feel like we can look at something more interesting
I have
than
a tick
spit
in my
though.
backpack.
Well,
You have a sample?
you have a tick?
Or you have a
Oh,
tick in
that's
your-
fine. Oh, this also isn't plugged.
I've got a
in.
seashell from Monterey yesterday.
Oh,
Actually.
that's cool.
There's this one
The
doesn't have
tick might
objective.
work. Yeah, if you want to throw this in
Nice.
there
Just calling
quick,
it in my
and
backpack.
then this should also have a
Hello,
power
am I audible?
No,
Hello. Yeah.
Shivam,
Yes,
hello?
yes. Yeah, I was
Hello.
just requesting like if you could like, when you are taking the names of the parts, like I'm not very aware. So if you could just define a little bit like, for example, maybe objective, like because objective for me may be used differently, but a friend clarified that. But maybe I'm like I myself is not very comfortable with the key terms of like, so if you
And
could just kind of
yeah, for sure.
define the
Yeah,
beginning that
Microsoft has a lot of
I
jargon.
think
So
everybody may not have used a microscope. Yeah, everybody may not have used a microscope. So it
yeah,
is
yes, I'm just kind of it's
yeah,
kind
and
of
this is an intense introduction to microscopy, I think. But yeah, so just let me know if there's anything that I don't define that doesn't make sense. So maybe just a little more basics while Kevin is screwing on this
Yeah.
objective here.
Yeah. So an objective. So in order to see something and magnify it,
I don't know what the
basically,
best is, sorry.
the objective is called an objective because it's the lens that's closest to the object of interest. So
Is that what it's called, though?
yeah, that's why it's called that. The other tube lens is because it's in the tube. Yeah. So there are many considerations about lenses. One parameter that specifies like lenses are different. One number that differentiates them is their focal length. A focal length kind of defines one important aspect of the lenses. If you have light that's coming in, like you can think of light as rays, right? If you have light that's coming in parallel to a lens, when it hits the lens, how will that light bend? And the focal length tells you that if you bring in parallel light and hits a lens, it will hit one focal length away. Likewise, if you put the object one focal length away, how do you think the light will come out? It will come out parallel. So there's this symmetry to how lenses work basically. If you look up any like optics diagram for a microscope, you'll see there's two major lenses. There's the objective lens, that one's the closest to the object, and it's usually positioned one focal length away from the object. So then if you think about an object, one focal length, lens. So now you have parallel light, right? You have parallel light, or people say collimated light. That parallel light will feed into the second lens, which is called the tube lens. The tube lens only sees parallel light. So then the tube lens will then focus the light again, and where we want it to focus is on the sensor of the camera. So you have object, lens, tube lens, camera. So those are like the four main parts, right? Sometimes you see like mirrors and stuff, but mirrors, if they're flat, don't actually bend the light other than redirect it. So if you can track those like four parts, like object, objective lens, tube lens, camera sensor, I think you can understand most of the basic of optics. So for example, here we have the objective lens, or the sample is going to sit here on this stage. Yeah, so the sample is going to sit on this stage. This is the objective lens. And I know it looks like pretty long and fancy, but you could essentially think of it as like one lens that's going to make light parallel. It's going to make light parallel all the way in here, and it's going to hit a tube lens. There's a tube lens, right?
There's probably a few.
Yeah, there's like a tube lens, and it's going to focus it on the center of the camera. So that's how, then you might ask yourself, like, where does the magnification come in? So actually, the ratio of focal lengths of the tube lens over the objective lens will give you the magnification. So it's some geometry that you can work out for why that is, but it has to do with similar triangles. Yeah, so that's like a very like quick rundown of like the main parts of a microscope and how the magnification happens. Yeah.
So in like a more general sense, right? You have your sample here, and then this whole column of stuff is basically just a series of lenses to make sure the light is focused and magnified at the point of the camera. So like all of microscopy is just figuring out how to get as much light as possible through the sample, and then as magnified as possible, but without like distorting or blurring the image. Yeah, so it's it's the gist of it.
We're gonna do your thing.
Yeah, so this is throwing a software error.
Can you do the main camera only on Octopi Research?
Is it a different folder? I just opened the folder that was opened already.
Okay. Let me try this one.
So Octopi's the incorrect plural.
Octopode is the correct one, right? Yeah.
Yes.
Yeah.
Let me try and make this.
Is there anyone that correctly said octopi?
It's not right.
So what did you check the
They
config
just-
file
they, like,
later?
Oh
I
my
don't know
God,
what
yeah,
camera. Okay.
Oh, it's
But this was, did you see this one working?
I
Or-
Greek, not Latin.
think
So it's like they
Okay.
think it's Latin. They think it'd be Latin, so they have to go Octopi. but actually-
Octo is a
Oh, this has
Yeah,
zero
Greek.
camera. Okay, we're not using
So
this one.
there's two. It's Octopodia / also octopuses are the two technical.
Y'all know that I've always seen two at the same time.
So right now, in order for the software to run, you need to make sure that the camera is put into the config file. And I think that's where the error is coming from, so I'm making sure the info from the camera is correct on the config file.
Any other, I'll field any other questions that you may have about terminology that he threw out. Okay, I was confused about what a gyro was many years ago. This is how I'll describe it. So a motor, let's focus on a type of driver specific, a motor driver. You think about what a motor is and how it works, there's a lot of physics that goes on. There's two, a stepper driver involves like configure, there's like 4 wires, like you had to like alternate pins like rapidly crazy and in a certain sequence, right, to make the gears like to make the magnets move relative to each other. And if you were to code that on like a computer to send signals to the motor, it'd be very tedious because it'd be like do this, then do this, then do this, then do this. So it's a lot of instruction. What I want to do as a programmer is just say take one step. So a driver is that middle person who is very knowledgeable about the device, but knows what the programmer is talking about. So the driver will be like, take one step, and then the driver will be like, I know what that means. It means they want to do a sequence like this. And if the programmer says, go faster, then like the driver knows like, oh, it means like I need to do some other hardware settings. So I like to think about the driver as the knowledgeable piece of code that sits between the like software, high level software, and the low level hardware. Yeah. So like when another example is like a camera driver. Like a camera is like a whole sensor array, and you have to do a lot of like have a lot of hardware knowledge to control this array. But the programmer is just gonna be like, take picture, and then right, a very crude instruction, and the driver will be like, oh, I know what that means. It means like they want to like do these things to this array and all this hardware level things. So it kind of provides a level of abstraction, so the programmer can do what they need to do without worrying about the hardware level detail. So that's what a driver. So they are your friends. You think about drivers when you say like, this is a cool piece of hardware. I want to control it with software. I need to find a driver.
And is it a physical component or a technical?
So
Software or is it a hardware?
that's a great question. I think my interpretation is that there are many levels of drivers in between. Sometimes they're a physical piece of like chip that makes controlling hardware easier, like a stepper driver. It's like you send electronic signals to a stepper driver and it'll do its thing. Sometimes they can sit a little higher, like in software land. But I would say it's like middleman. That's like there's many because like there's two ends, but there's a lot of middle. There could be a lot of layers of drivers. Right, Ethan? Yes. Yeah. There could be a hardware driver or like a driver for the software to hardware. That's the part. Yeah.
We're missing a power cord.
So here it's basically supposed to control the motor.
Yeah, so there's a motor driver, and there's a this board controls the motors. The camera is controlled directly by the computer, so the camera does not go through this board.
Can you explain that gold piece he was talking about and how that lets you?
So do people know what a piezo is? Okay, yeah. It's not like in the common knowledge of a layperson.
Not, yeah, I don't think.
I sort of know what it is, but not like I just know.
Yeah.
So I think piezo, maybe you know the root of it. It means like pressure or something.
No, I don't. I'm only in the marine space.
Okay. So piezo is a piece of like cool material that has a nice property. If you apply a certain electric voltage to it, it will like change shape. Essentially, it'll change shape in a very specific way. So likewise, you could do it the other way. If you like push on the piezo, it can actually generate electricity. So, but in this sense, we want to put in electricity to change its shape. So one way you can change its shape is to like make it taller or make it bigger, right? So that has the effect of like, because the motor is like moving things up and down, but the piezo it's like moving down, up and down, very tiny, based on the electrical signal you're putting into it. So that gives you fine control over the focus, because at the end of the day, how far does the how far does the objective lens need to be from the object? It needs to be a focal length, right? So to get it precisely, you need big control, like big control, but you also need tiny control, right? So that's what a piezoelectric is, and that's what this object is, and why there are wires coming out of it.
And he was saying that extra material sounds incredibly complex is somewhat straightforward and really
Usually quartz crystal.
Yeah.
So like the digital watches, those are based on quartz crystal.
oh,
Yeah.
fantastic.
Yeah,
That's
they
really-
oscillate. Yeah. I think there's some ceramic ones too.
I think so.
Yeah.
I think the ones that you'd be using for focusing your phones.
Okay. Yeah. So sorry, what was your question?
Oh, I was just saying that sounds like sounds like a crazy material, but I guess it without knowing what it is, but I guess it just sort of.
It's pretty ubiquitous. If you think about how electric guitars work, they use piezoelectric.
Oh.
Like if you ever played an unplugged guitar, it's very quiet. But because the strings vibrate, it actually vibrates a piece of piezoelectric. So this is an example of a piezoelectric going from mechanical to electrical, because the mechanical vibration of the string generates an electrical signal which then feeds into the amplifier. Yeah, blows your mind, yes. Yeah. What's another example?
So this is filtering software.
Okay, okay.
Ethan, how close are you to being done? Otherwise.
If you wanna wrap up, take as much time.
Yeah,
Oh no, this isn't working yet.
yeah,
It looks great.
yeah,
Yeah.
we learned a lot about the hardware, at least today.
They can also do fluorescence. Cause I was confused, how do you do fluorescence?
Can we use this as
So
an
the-
example?
Great question.
We just look through it like that.
So, yeah, he
So,
was
you,
talking
so
about
this
the
is
older
a full
of models that exist.
setup, right?
Uh,
We
this
just
is
can't
one
visualize it on the screen right now.
or
Is the
exciting.
um.
This would just be bright field or dark field, depending on what's.
So you actually can, so
Other way.
for more general sense, so a lot of times if you're interested in looking at like certain aspects of biology, like you want to label a protein or you want to look for like a fluorescent signal, or like bioluminescent signal that just occurs naturally, we can focus on certain wavelengths that excite like fluorophores or things that basically light up just under certain colors or wavelengths of light, and then look at them using fluorescence microscopy, is what that's called. And so to do that, you're basically just filtering out which types of wavelengths of light, like out of the spectrum of light colors, hits your sample, and then which wavelengths you're actually detecting. So instead of detecting like all white light, you're just detecting like red light or something like that. So to do that, we can actually have different versions of this optical train that will excite, it'll be like epifluorescence equivalent. So it'll excite a certain color or wavelength of light, and then have like a mirror that filters out the other things called like a dichroic mirror. And then you'll be able to look at it that way. So there's a few versions of that. We haven't used them as much, I think, on SQUID, but it's definitely possible just by changing out this optical part. And you can still use like a monochrome camera to be able to detect that.
Peeping at the dick.
Nice.
Yeah.
Which
Does
is
it work
in the
that way?
magnifying glass. Where'd you get the tick?
Me.
Okay, that's fun.
He wanted to be friends, but I
You turned him down.
not looking for the same kind. Her.
Yeah,
You
you
wanna
want to
do
talk a
so?
little about the pricing stuff? Mother has mentioned it a bit, but this is the first time they're seeing it in person. If you guys wanna like, come closer to see.
Yeah. So this is the Plaktoscope. It is a microscope for imaging particles present in water samples. It was developed in our lab a few years before I joined. So I don't know, like 2016-ish, probably. Yes, so the idea is this is a low-cost microscope that's aimed at kind of providing similar capabilities as existing microscopes, which cost closer to $10,000 or $100,000. But this is like several hundred to a few $1,000s. And so the idea is basically we have a water sample with plankton inside or microplastics or other suspended things. It goes into this capillary tube that all the particles flow through as they get sucked through this tubing by this pump. So that's just a peristaltic pump. And so as the pump pulls things down the tube, the particles pass through this glass capillary tube, which we call a flow cell. And we have a camera that basically can watch everything that passes through. So this is a water sample from Searsville Lake in Jasper Ridge Biological Preserve, like a 10-minute drive from here. And basically yesterday, me and a rotation student went out to the lake and we just used a syringe to pull up water with pond scum. So there's often interesting things living on or near the pond scum. Yeah. Oh. Yeah, there are lots of things that look pretty dead. Occasionally, you might see something swimming around. Yeah, there's some
Like swim back.
You
You ever see any tardigrades or like water bears?
I don't think we've seen tardigrades in these samples.
What magnification is this right now?
I don't remember the exact number, but this usually is good for imaging particles from around 40 microns to 200 microns.
Wow.
Anything bigger and the particles kind of get clogged. Anything smaller and they don't really get high resolution images in the optics that we have right now.
You have to filter before you put it in.
Yeah, exactly. So this is a filter. It's basically a mesh here. So we just pour the water in, the big things get stuck, and then the small things go through.
Would the pond scum clog it up again?
Yeah, so I think most of the pond scum either sunk in the bottom of the bottle before I was able to pour it in, or it got trapped by the filter. Yeah. Oh yeah,
That's
something.
something.
Let me slow down the flow rate a bit.
Didn't you start a stream about this? Wasn't there a stream of Plaktoscope before?
Oh yes, I did an experiment where I was live streaming the video feed from here onto YouTube, just because Manu was very excited about doing that.
Do people watch?
Some people from our lab saw bits of it, I think. Yeah,
On YouTube or you not?
on YouTube.
Should be
Yeah.
Twitch.
Yeah,
maybe.
like,
We
what's
also did
that?
Twitch because I had problems with YouTube's streaming infrastructure for a bit. But yeah,
What how do you do if like there's something that you want or that you are looking for that comes across the screen?
right.
How do you
So right now, the way the Plaktoscope acquires data, so right now we're kind of in preview mode, and then there's data acquisition mode. So the way the Plaktoscope does data acquisition is that basically, if things are moving in front of the camera, then they kind of become blurry in the images that we save. So what they did to kind of simplify this and keep the hardware simple, avoid the need for fancy hardware stuff, is basically the in data acquisition. Actually, let me demo this maybe.
Sweet.
I change the name of the project.
This is a laser cut or is it wood?
I think
machine?
it's CNC milled, and this is bamboo plywood flooring or something. Is what I heard. Yeah, so you can treat it to make it waterproof, but I don't think this has the treatment. Okay, so now I'm starting data acquisition. So what it does is basically it has the pump pull some fluid, stop, and then take an image. Then pump some more fluid, stops, takes an image, and so on. So here we're kind of really only imaging maybe 10% of the particles in the sample. But this is how we get nice high resolution images that are not so blurry because things usually don't move around. Although sometimes things are swimming, as you can see. Yeah, so that's kind of what we have right now. What we'd like to do is move more towards something where we can maybe image everything, but that's going to take a lot more work on the computer vision algorithms for detecting and isolating objects so that they can do it in real time instead of doing it after the fact. Yeah.
And you have the machine learning model like running on the images and identifying the objects?
So I think people have been doing work to do that. Usually right now what happens is we collect the whole data set and then we pass it afterwards into whatever scripts people have made for doing whatever they want to do.
Do you think you could tell a difference, like from your eye, if it's a freshwater sample versus a saltwater sample?
Oh, that's a good question. I think often saltwater samples will have different kinds of plankton that will show up, but also that really depends on where your saltwater samples are coming from. I think with freshwater samples, I think it also really depends on how you collect the samples, because we got 3 samples from the same place in the lake. One was kind of at the surface where there was pond scum. One was from the surface where it was like clear water, and then one was from the bottom where there was like the sediment mud layer. This has almost nothing inside it. And it looks very similar to when you just get seawater and there's almost nothing inside it also. Yeah. So what they do with seawater samples, usually they'll have this giant net with a mesh that's a very fine mesh. And then there's like this bottle at the bottom. And then they basically filter water like a whale, basically, to catch all the plankton. Yeah,
Cool.
so those are plankton nets. Oh. And then that's a way of concentrating the samples so that we don't just see empty stuff most of the time.
Ethan, can you hear me?
Ethan, I think you have a question from Zoom.
Oh, yeah.
Yeah, could you explain how the segmentation works in this? Is that AI based or like how does that work exactly?
Yeah, so what exists right now is not very sophisticated. Basically, it's a Python program which uses OpenCV. So it does like some background subtraction because the image here is kind of dirty right now. I need to clean the flow cell, but you see there's like stuff that is stuck. This is like dust or dirt on the outside of the glass capillary tube. So basically the script estimates the background, subtracts it out from the images. And then what we're left with is kind of something that we can apply a threshold to. And then there's some operations. It's like expand, dilate operations to try to merge adjacent objects. And then there's a thresholding at the end. And those are the masks that are used to say, this is object 1, this is object 2, this is object 3, and so on. So it's not sophisticated at all. It was also written before I joined the project. And I think they want to rewrite the thing, but I'm not involved in that. But what we have is like this, when the data gets collected, it basically saves a series of these raw frames. And then you can do whatever you want with them if you don't like the existing segmentation algorithm.
Understood.
Excuse me.
Yeah. And one more question which I had was that like since the capillaries are thin, how do you make sure that you're focusing inside the capillary rather than on the back of it or like the front side?
Right. So with the current optics and the current capillary, there are like small objects are going to be out of focus much of the time. This is why we don't recommend using thicker capillaries with the current optics because the depth of field for the focusing is still the same. So more objects are going to be out of focus. It's something that you just need to collect enough raw images that you have enough objects that you have good statistics on things that are in focus. I think the company that makes these microscopes now, they are working on providing different combinations of lenses that can work better with thicker capillaries if you want to image larger objects like copepods.
Excuse me, can I ask a question? So, is there actually a machine learning model to identify the planktons?
Right. So I think right now the Plaktoscope software itself, it basically ends its work at extracting objects, and then you can do whatever you want with those extracted objects. Right now, the most common use case is that people will upload these extracted objects to a website called Ecotaxa. This is a project from a French marine lab. Oh, because I'm connected to the Plaktoscope's Wi-Fi hotspot.
I can look it up.
But if you can, Ecotaxa and screen share.
Yeah.
So Ecotaxa is basically a web interface / image database that provides an easier user interface for scientists to label all of the objects that they have extracted from a Plaktoscope or one of the fancier instruments that exist. And also provide some tools to like make predictions on objects. Actually, let me send you a link, maybe.
Which one is this? Not the right.
I want to send you a link to a specific project.
Okay.
Okay, maybe if you just do explore images, and then it'll take you to a random project.
Wait, do I think it's cute or do I think it's yours? Yes, and yes, I would say that.
You can search a project.
Try refreshing. This looks like a useless random project.
This one?
Just refresh the page. Okay, I don't see anything there. Refresh again.
Hell yeah. Got any slides?
This looks, yeah. So these are objects that were extracted from some other instruments. So your screen sharing, great.
Yeah.
Yes, okay. And so this Ecotext interface basically allows you to manually label objects, and then you can use a data set, for example, the objects that you've already manually labeled, to run predictions on unlabeled objects. And then you can go back and check and confirm the predictions, or correct any incorrect predictions. So that's the machine learning capabilities that currently exist and are currently kind of commonly used with the Plaktoscope. There are also scientists who have developed their own analysis scripts that might do other things with automated classification.
So yeah. So sorry. So basically, the Plaktoscope, when you extract the images from the Plaktoscope, you will upload it in this website to make the identification.
Right. That's the most common way that people use images from the Plaktoscope is they upload it to this website. Yeah.
So it would be better to implement, let's say, a machine learning model directly in the Plaktoscope and do the identification
I would
directly.
recommend against that because the computer in the Plaktoscope is Raspberry Pi, so it's extremely not very powerful for doing machine stuff. I think you could optimize a model to run on the Raspberry Pi, maybe with the hardware accelerator for machine learning models. But I think that's a lot of work, and it's probably not the kind of thing you would do at the start of a project. This would be after you've developed a model, then you want to transfer it to run in real time on the Plaktoscope or something. Yeah. Right now, like the segmentation program that runs on the Plaktoscope is also pretty slow. So people have been looking at running it on their own computers. That's what I do for data analysis. Yeah, I would recommend avoiding running computationally expensive algorithms on the Plaktoscope unless you absolutely have to.
Yes, you may implement a cloud service that contain a machine learning model. Though it will be, I think, not that expensive. I don't know, just
Yeah, you could do that. Or even if you don't have internet access, you could just provide your own computer that has the model and you do the data analysis after transferring it to that computer.
Thanks a lot.
Awesome. Anyone else have questions about the Plaktoscope?
Yeah, I have one actually. So how do you clean it after use so that you know?
Yeah, so basically what we do is we flush all of our sample out from the Plaktoscope fluidic stuff, and then we rinse with clean water. And then usually that's enough if you just need to clean it between samples. But stuff does get stuck into the walls of the glass capillary. So you do need to routinely run bleach through it to kind of kill all the stuff that's stuck and try to neutralize the sticky substances. So you flush with bleach for a while, and then you rinse with clean water again, and then usually that's good enough. Sometimes you also need to use some special paper or cleaning fluid to clean the outsides of these glass capillary tubes. But that's about it. I see another question.
Yeah, Debbie Yodi.
Yeah, hi. So I just want to know, like, can you use this Plaktoscope to, measure and find the distribution of microplastics, their sizes and something like that? Can it be used for that?
Yeah, so I think people have been trying to do that. I don't know what the status of their projects are. The main thing is that you would need to select microplastics in a size range that the Plaktoscope is able to image. But I think you can do that. In some of the marine samples that I've processed, I've seen microplastic particles, although not in large enough quantities for me to really quantify that.
Okay. Thanks.
Yeah, there's a lot of interest in using it for microplastic stuff.
The water just going back into the sea.
Yeah, just because I don't want to waste sample. But usually what you would do is you would put it in the waste tube. Yeah. Because the pump crushes stuff that passes through because it's a peristaltic pump. It doesn't crush everything, but it crushes enough things that we want to be.
All right. I think Anna, how is this one going?
Something, but I'm not sure how Enrico.
This is not actually Enrico's. Enrico saying it's Enrico's, but it's not.
Okay, but I'm still not sure how. Like, I don't think the Z is moving.
Oh.
Because it's connected to this, and this is like bolted in.
Yeah.
Then it should be usually the optical. So the construction of this is usually where you have the optical chain. So this is that objective with mirrors and the camera is attached to this Z stage. So this is what makes the objective move up and down to focus. But it looks like this was attached to this piece, which is like kind of bolted in. So it's not free to move up and down as far as I can tell. Right?
Really small.
That's not moving at
Yeah,
all.
don't sweat it. Don't sweat it. If anything, can't you just like you do a manual focus like this?
So I'm gonna try to unscrew this and just get. the
Yeah,
objective
can't
back.
you move this one up and down?
It's just a bit, I'll
Like actually surprisingly. Like, we should try one of these times. We should try.
Kevin, I have
Yes.
yeah. Could you tell if we could change the magnification of the Plaktoscope?
Like monorail.
Yeah, maybe that's a question for Ethan.
So you could, in theory, change out the lenses. But I think you would need to know a bit about optics to choose the right lenses. And I don't know enough about that myself.
Like this one, wet.
It's an open source project or open hardware project, so you can make your own Plaktoscope to play with as well.
Yes, it's a
Yeah.
bit complicated to make everything yourself.
Yeah, I would honestly probably take time to find it and do it.
I have one actually. I was just I
And
Oh.
just find it and be like, oh,
Awesome.
because otherwise I don't want to do that and then not find
Yeah.
it. That'd be lame.
It's cool. Let's see. I think we might be wrapping up in about 5 or so minutes. It's almost 12 here. Oh, did you see? Oh my gosh, we got something. Oh my gosh.
So this is a tick that we just taped over where we couldn't find a slide. So these are each legs of the tick, and then this is its body right here. And I can move, I think X, Y is.
Okay. You guys want to see a tick? Is that the head?
There is.
Oh my god.
Let's see if we saturate this, if we can get
There
it
is. Look at that little proboscis.
from backpack. Or was it on your backpack?
It was just on my back.
What are you trying to do?
If there's any like semi-transparent parts, sometimes if you saturate them, you can see, but I don't think there's much there. But yeah, so these are the legs.
Whoa, we get
That's the head. A joystick. Oh yeah, it's very much like a game controller setup. There's a cool looking leg.
Jack's blood in there, by chance?
Oh, maybe.
Not given willingly.
Any insect people? Any insect can we tell if it's something?
My assumption is that with most of the.
Oh, is that the limit already?
Females are the one that go.
I bet the males, yeah, probably. Super teeny on its side.
Legs. That's a great question. Right?
It should it's back. I think we put it back down.
Sounds right to me.
Can I try this?
Yeah, please.
So right now the focus isn't working, but the left right and up down movement is working.
I think there's a limit to how far it can move.
You can see that the
I think it's male.
the helix, like the drive when he was talking about, is moving.
Oh, there you
when
go.
you go left and right.
I think you can literally see that
Yeah.
bottom curve at the back of it.
It makes sense to me. Does it say whether, well, we can Google it, I suppose, whether males take blood meals?
If you want to go to a group meeting, I think I can help clean up from here, or this section at least. Yeah.
Yep.
Okay. All right.
I'm glad to see the head is intact.
Yeah.
Yeah.
Luckily.
Ethan, I think we
you
should
make
be
a
good,
slide.
yeah.
Do you make a slide just with tape?
The so is a motorized stage sufficiently expensive to, I'm sure it is quite expensive to justify like designing and making all these parts.
Do you have like stage like from a company?
Yeah, I guess something like that.
The price point does go up.
I'm sure.
Like an order of magnitude.
How,
I think there's a bunch of different kind of like open source methods that people
Yeah.
have used to make similar stuff.
Cause I really
Um,
wanted
did they get to
basically this.
What?
for
Yeah.
a
They get
number
to
of
see this?
Yeah, they
years.
did.
But
Um,
it
how would this work with 3D
not
stuff?
really
Would
felt
you find
justified
it?
to like take it.
They do have composite modules.
yeah, I need to check.
Like,
Like,
a lot
literally,
of
like, if I had
modules
my
would be
little
very
coral
different.
things.
That is also true. I mean, it's always, it's always
This
tough.
still works.
Like
Yeah.
Howard
So
just
for
buys
3D stuff, like
as I
as you can
like
see, it's
sometimes
imaging
when
from
I
below,
spend
but the
some
light
time,
is
like,
from
tinkering
above,
with
so it needs to be
something, like, trying
translucent
to think
enough.
of some method
So
to
different
try to, like,
samples
yeah, I could
are
just
difficult.
buy a
So, like,
dozen of them. Out.
That probably wouldn't work.
But yeah. but
No,
so a
what
lot
you
of
would
them do
need
have
is
things
like
A shade.
something that shines from below.
Which is something that's so cool.
Yeah, so you could move the light source, and but you only be able to study
Whose lab were you in for your
surface of
PhD?
it.
Oh, I'd only
Got it.
be looking at stuff.
Yeah, okay.
Oh, I see.
Yeah, I think you would need to maybe move the light source over from the bottom. Yeah.
Okay, interesting. Or you can take like a tissue sample of it off the top and then use that on the
Yeah,
slide.
exactly.
This is really cool.
Yeah, thank you all so much.
Yeah.
It's awesome.
All right. You else want to play?
Really clap.
Yeah. All right. Thanks for coming.
Sweet. Thank you.
Thank you everybody for joining in on Zoom.
Um,
Yeah, I'm gonna end recording.
I am interested.